Hello everyone. So today I'm going to be talking about the explanation for topic 7C, control of the internal environment. Now this is the last chapter from our entire topic 7. So let's begin. Okay, so the first slide is the structure of kidney. Here is a diagram of a kidney, and now let's look at the labeling of the kidney. There is the fibrous capsule. Then there is the cortical nephron, which is the loop which only just enters the medulla. There is a cortex. This has a particularly rich capillary network and is very dark and red in color. It contains the Malpighian bodies of all nephrons. And then there is the medulla. This contains the loop of Henle from the nephron, the pelvis, center chamber where urine arrives from the collecting ducts, juxtamedullary next to the medulla nephron. The long loop penetrates right through this medulla. Pyramid, a collection of tubules collecting ducts and blood vessels, and the ureter, which carries urine down to the bladder. Okay, moving on to the next slide. What is homeostasis? First, let's define homeostasis. Homeostasis refers to the body's ability to maintain a stable internal environment despite any changes in the external condition. It's all about balance. So, keeping factors like temperature, pH, glucose levels, and water concentration within precise limits. During exercise, our muscles produce more heat, which could potentially disrupt this balance. This is where homeostasis steps in, using mechanisms like sweating and increased blood flow to cool us down, keeping our body temperature in check. This continuous adjustment process is known as dynamic equilibrium. This allows the body to adapt to challenges while staying stable. What is the role of a hypothalamus in thermoregulation? The hypothalamus plays a central role in this process. It acts like a thermostat in the brain, constantly monitoring the temperature of our blood. When it senses that our core temperature is rising, it triggers responses like sweating to help dissipate heat. Conversely, if it detects a drop in temperature, it initiates shivering and vasoconstriction, which is the narrowing of the blood vessels, and this helps to conserve heat. Moving on, the production of urea from excess amino acid is part of the process of deamination. So let's look at the steps for deamination. First, step 1 is deamination, which is excess amino acids cannot be stored in the body. So they are broken down in the liver through a process called deamination. In this process, what happens? The amino group, which is the NH2 group, is removed from the amino acids. This amino group is then converted into ammonia, NH3, which is highly toxic for our body. The general equation for this deamination is amino acid plus oxygen gives us keto acid and NH3 ammonia. The keto acid is the remaining part of the amino acid and can be used in respiration or converted into glucose or fat for storage. Okay. Now, second step is conversion of ammonia to urea. Because ammonia is highly toxic, the liver actually converts it into a less toxic substance known as urea. This is done through the ornithine cycle, which is also known as the urea cycle. This cycle involves a series of enzyme controlled reactions where ammonia combines with carbon dioxide to form carbamyl phosphate. Carbamyl phosphate enters the urea cycle and eventually leads to the production of urea. So the simplified reaction in the urea cycle are ammonia plus carbon dioxide gives us carbamyl phosphate. Through several steps, this leads to the formation of urea, which is then transported via the kidney to the by the blood to the kidney. Sorry for the mistake. Urea is This is step 3, excretion of urea. Urea is transported from the liver to the kidneys in the bloodstream. It is excreted from the body as part of urine. So, this process allows the body to safely remove excess nitrogen from the breakdown of amino acids, ensuring that ammonia does not accumulate to toxic levels. And this urea cycle is crucial for nitrogen waste management in the body and is essential for part of ammonia acid amino acid metabolism. Moving on to the next slide, now let's talk about the kidneys and their incredible filtering power. Blood enters the glomerulus of the nephron at high pressure via the efferent arteriole. This high pressure forces small molecules such as water, glucose, ions, and urea through the capillary walls into the Bowman's capsule. But larger molecules like red blood cells and plasma protein remain in the bloodstream because they're too big to pass through. So the small molecules also passes through the capillary to the basement membrane, the Bowman's capsule. Okay, to mention the basement membrane as well. This initial filtration process ensures that only waste and small molecules move into the nephron, setting the stage for the next steps in urine formation. So, also remember that larger molecules like red blood cells and plasma proteins cannot pass through, but they will be reabsorbed back into the bloodstream and they will return back to the bloodstream via the efferent arteriole. So now we speak about the selective reabsorption in the nephron. Selective reabsorption primarily occurs in the proximal convoluted tubule, where essential substances like glucose, amino acids, and ions are reabsorbed back into the bloodstream, remember, via the efferent arteriole. This step is crucial because it prevents valuable nutrients from being lost in the urine. The reabsorption of these substances happens through both active transport, which requires energy, and passive transport, like osmosis, which does not. This precise control allows the body to maintain the right balance of fluids and electrolytes, which is vital for normal cell function. This is the next part of the selective reabsorption, but this is the role of loop of Henle in reabsorbing water as well as the countercurrent multiplier theory. Now this is a very important concept and please pay attention what I'm about to say next. So what is the role of loop of Henle in reabsorbing water? The loop of Henle is a critical structure in the kidney that plays a critical role in concentrating urine and conserving water. It's a U-shaped loop found within the nephron and it consists of two limbs. First is the descending limb, another is the ascending limb. These limbs have different permeability properties that are essential for the reabsorption of water. So, what is the function of the descending limb? The descending limb is permeable to water but impermeable to ions such as the sodium and chloride ions. As the filtrate moves down the descending limb, water is drawn out by osmosis into the surrounding medullary interstitial fluid, which is highly concentrated with ions. This movement of water in the surrounding tissue increases the concentration of the filtrate as it moves towards the bottom of the loop. Then there is the ascending limb. The ascending limb is impermeable to water but absolutely permeable to ions. As the filtrate moves up the ascending limb, sodium and chloride ions are actively transported out of the filtrate into the surrounding medullary interstitial fluid. Since water cannot follow, the filtrate becomes progressively more dilute as it moves towards the top of the ascending limb. The removal of ions contributes to the high concentration of solutes in the surrounding tissue, which helps maintain the concentration gradient required for water reabsorption. This selective permeability and transport of ions create a concentration gradient in the surrounding medulla, which is essential for water reabsorption later in the collecting duct. Okay, now we move on to the counter current multiplier theory. The counter current multiplier system is a mechanism that allows the loop of Henry to concentrate solutes in the medulla, creating a steep osmotic gradient that helps conserve water. Counter current refers to the flow of filtrate in opposite direction in the ascending and descending limb of the loop of Henry. Multiplier refers to the ability of the system to multiply the concentration differences established by the movement of water and ions. How the countercurrent multiplier works: in the ascending limb, water is drawn out by osmosis because the surrounding medullary interstitial fluid is more concentrated with ions. This makes the filtrate inside the descending limb more concentration as it reaches the bottom of the loop. In the ascending limb, sodium and chloride ions are actively pumped out of the filtrate into the medulla, increasing the osmotic concentration of the surrounding tissue but making the filtrate more dilute. The pumping of ions creates a high concentration of solutes in the interstitial fluid surrounding the loop of Henle, particularly in the medulla. This sets up the conditions for water reabsorption in the collecting duct. The concentration gradient. The longer the loop of Henle, the greater the concentration gradient in the medulla, which results in more effective water reabsorption. This gradient allows the kidney to produce concentrated urine when the body needs to conserve water. So, this is it. Please rewatch this segment if you feel you couldn't have catch enough so that the concepts are clear in your mind. Now let's move on to the role of ADH in water regulation. Antidiuretic hormone ADH is crucial for regulating water balance in the body. This ADH is produced by the pituitary gland. That ADH controls the permeability of the distal convoluted tubule and the collecting duct in the nephron. When the body is dehydrated, more ADH is released, making the walls of these structures more permeable to water. As a result, more water is reabsorbed back into the bloodstream and the urine becomes highly concentrated. If the body has too much water, ADH levels drop, making the ducts less permeable. This leads to less water being reabsorbed and the production of dilute urine acid. This precise regulation of water ensures that our bodies can respond quickly to changes in hydration status and maintain homeostasis. So, this is one slide that is made entirely from the book content, and it's an important thing to be learned. This is one of the most important questions from this chapter as well. That is the mechanism of the ADH action. What is the mechanism of ADH action? The mechanism by which ADH increases the permeability of water of the cells of the distal convoluted tubule and the collecting duct is very elegant. ADH does not cross the membrane of the tubule cells. It binds to specific receptors which triggers reaction that result in the formation of CAMP as the second messenger. The CAMP starts a reaction of series of reactions that causes vesicle within the cells lining in the tubule to move up to and fuse with the other cell membranes. The vesicle contains water channels which are inserted into the membrane and make it permeable to water. Water then moves through the channels out of the tubules and into the surrounding blood capillaries by osmosis. The amount of ADH released control the number of channels that are inserted. This means that the permeability of the tubules can be very closely controlled to match the water demands of the body. When ADH levels fall, levels of CAMP also drop, and the water channels are taken out of the membranes and repackaged in vesicles. This makes the tubule impermeable to water again. The channels are stored in vesicles, ready to reuse when they are needed again. Please give a thorough read to this section of the mechanism of ADHD action, and please take notes from your book and read the book very, very carefully. So, lastly, let's explore how our bodies control which genes are active at any given time. This regulation involves transcription factors, protein that binds to specific DNA sequences, either turn genes on or off. Remember, this concept is from your Unit 2 of the IAS course, and that knowledge might be linked to this knowledge that can relate to questions that will come in the exams. So now, the peptide hormones. These hormones, like insulin, act on receptors on the cell surface since they cannot cross the cell membrane. When they bind to these receptors, they trigger a signaling cascade that activates the transcription factors to regulate gene expression inside the cell. There is the steroid hormones. In contrast, steroid hormones like estrogen are lipid soluble and can pass directly through the cell membrane. Once inside, they bind to intracellular receptors, forming a complex that directly influences gene transcription. By binding to DNA in the nucleus, this ability to switch genes on or off allows cells to respond precisely to their environment, whether it's managing growth, metabolism, or other vital functionings. And that's a wrap on our in depth look at how our bodies control their internal temperature. If you find this video helpful, please let me know. If you need any help or if you have any questions, you can ask me at my mail or any other place. So, thanks for watching, and I'll be back with another video. Thank you for listening so far.